In 1957, a young biologist killed an entire family of theories about the genetic code. He didn't run an experiment. He never touched a test tube. He used a pencil and a handful of facts everyone already had. The double helix was brand new. Watson and Crick, 1953. Everyone agreed DNA carried the message, but how 4 letters spell the 20 amino acids of a protein, even how the protein got built, was wide open. There was no known messenger, no adapter. Many simply assumed the amino acids lined up against the DNA itself. The arithmetic teased. Pairs of letters give 16 combinations, too few. Triplets give 64, more than enough. The code wasn't yet a job for experiments. It was a playground for theorists with pencils. And the most beautiful guess came from George Gamow. With no messenger known, he reached for the most direct idea imaginable: build the protein right on the DNA. Each amino acid would drop into a diamond-shaped pocket along the double helix. Look closely, and each diamond is really a triplet. 4 corners, but 2 are a base pair, A with T, G with C, so they count as a single letter. 3 letters read in overlapping steps. And by a thrilling coincidence, the diamonds came out to exactly 20 shapes for the 20 amino acids. It was beautiful. The only question was whether it was true. So picture the DNA as a long tape of letters. A non-overlapping code reads it in separate blocks of 3. An overlapping code is greedier. It slides along just 1 letter at a time. Letters 1 to 3 are the first amino acid, 2 to 4 the next, 3 to 5 the next. Read that way, every amino acid shares 2 of its 3 letters with the one beside it. Here is the catch. Fix one spelling of an amino acid. Its 3 letters are set. The next amino acid reuses its last 2 letters and adds only 1 new one. Only the last letter is free, and a letter has 4 values. So any single spelling can be followed by just 4 different amino acids. Now for the empirical part. Brenner turned to the proteins chemists had actually sequenced. Above all, Fred Sanger's insulin, the first protein ever read letter by letter. He went along each sequence and noted every neighbor, which amino acid follows which. Take leucine. In insulin, it is followed by cysteine, valine, and tyrosine. Pull in the other known proteins and its list of neighbors grows to 7. And there is the collision. In the real proteins, leucine is followed by 7 different amino acids, but a single spelling allows only 4. So leucine cannot be one triplet. It needs at least 2. Now add it up. Every amino acid, like leucine, needs more than 1 triplet to cover all its neighbors. Brenner counted them across every protein sequenced by 1957, and the total climbed past 70. But a 3-letter code has only 64 words in all. 70 cannot fit inside 64. No overlapping triplet code can spell real proteins. So why does a non-overlapping code escape the trap? Because separate blocks of 3 share no letters at all. Nothing ties one amino acid to the next. Any amino acid can follow any other. So each one needs just a single triplet, however many neighbors it has. 20 amino acids, 20 triplets, and 64 to spend. It fits, with room to spare. And the verdict is sweeping. The paper's title says it all: the impossibility of all overlapping triplet codes, not just Gamow's, but the whole class, ruled out by a single count. The code had to be non-overlapping, exactly as a famous experiment would confirm in 1961. What makes it beautiful is how little it took. No apparatus, no new measurement, just a sharp question, other people's data, and arithmetic. Brenner didn't find the code. He cleared the board of everything it could not be, which is very often how the largest questions get answered. Sometimes, the most powerful instrument in the lab is a pencil. Beautiful experiments.